Dave Seftel reporting from Macular News from Arvo 2014. We're here with Ygal Rotenstreich from Israel, and he's going to tell us about some of the extraordinary innovations that he's been involved in developing, not only for macular degeneration, but for other eye diseases. Welcome, Professor. Tell us about your background, firstly.
Well, I'm working in the Sheba Medical Center. This is the largest center in Israel, and we are affiliated with the Tel Aviv University. And since that, we are doing clinical studies, research studies, basic research, and translational studies. That's how we combine all together in this big institution.
So you're both a basic science researcher and a clinician, is that correct?
Yes, I have a team, of course. I cannot work by myself. But yes, we have PhDs, MDs, everybody is there. Even physics and science from all over the areas.
So you have a multidisciplinary team working on a number of different innovations. Firstly, tell about your work in macular degeneration and the paper that you presented in that area.
We succeeded to develop a new technology, a new system, which we published recently, how to inject or how to transplant stem cells into the back of the eye. This is definitely very important in treating macular degeneration. With this system, the results showed that we don't do any retinal detachment on one hand, and on the other hand, we are getting the stem cells as a thin layer all over the retina, spreading it all. Now, another issue is that we don't go down to the macula. We inject the cells in the anterior part of the sclera, so it's only around 5 millimeters posterior to the limbus. So we're relatively very safe in this method.
Well, that's clearly a major change to the way it's been done currently.
Right. The way it's been done currently, for example, for genetic treatment and for stem cells treatment, is that we do need 3-port pars-plana vitrectomy, and then we do a bulb of retinal detachment. With this system, we don't do the bulb, we don't do the vitrectomy, and we are having all the retina treated. So it's, to my understanding, it's more safe and more efficient.
That's clearly a major advance, because technically it seems like your approach would be less arduous and less prone to risk of damage. Is that correct?
Exactly. We are now getting, after we did the study and published on RCS rats, which is a known model for AMD and Retinitis pigmentosa, we went and showed this in this ARVO in rabbits, which is almost a human eye. And then we are going further on in more expensive model called the pig eye. And from that point, we'll definitely go into the human eye, which is my interest as a medical doctor.
What is the time course for that?
The time course, I believe within a year we can go further in. It's of course a matter of budget, but if I had all the budget I need, a year would be enough.
So you expect to get an IND and regulatory approval to proceed with a clinical trial within a year to 18 months?
Exactly. That's what we are not holding back. We're just going further as much as we can so we can get the result done and have a better vision for our patients.
How did your team and you come to this particular invention, this approach?
I really don't know the real answer, but probably it's something in your head, how you start with things and it develop and suddenly it happens. It's not always that I know why it happened, but it is.
Well, thank you. It's clearly a major advance and one that we'll be watching very closely as it goes forward into clinical trials. Switching gears now, tell us about some of your other work in ophthalmology.
Yes, since we are treating patients, we need to diagnose them and we need to follow them up in clinical trials. One of the major limitations is that we need a visual field which is not subjective. All we have up till now is a subjective visual field, and this is a very problematic issue because the patients, whatever you give them something, especially patients who doesn't see very well or well, whatever you give them, they feel that it's getting them better. And if you do a subjective test, especially in the periphery, they feel it's getting better. And this is a hard way. This makes it hard to show the improvement, whether you have it in clinical trials. Because of that, we succeeded to invent a new way to measure the objectively the visual field. Not just objectively, but since we are doing it with a red and blue stimulus chromatically, we can distinguish between different photoreceptors. There are photoreceptors who are more responsive to the red light, so we are stimulating them by the red light. And there are other photoreceptors who are more responsive to the blue light, and that's the rods, and that's how we can measure those. Segregating those systems help us to overcome one of the problems of doing visual field with a white stimulus, because with a white stimulus you're averaging them both. And for example, if the rods are working but the cones doesn't, so the patient can still see the white stimulus, maybe a little less, but then you hardly can distinguish whether it's working or not. So this is one of the advantage of using this two type of stimulus. Another advantage is that in the recent studies, it has been observed that there are ganglion cells that are responsive to light. There are specific ganglion cells that are responsive, does not need a photoreceptor to be responsive. And if you shine a bright blue light, you can get their pupil response to those one. With our system, we can map those ganglion cells, which are particularly important in circadian rhythm, for example, insomnia and things like that.
Well, that's clearly another very, very useful tool in an adjunct to be able to remove subjectivity from the patient analysis.
Exactly.
Because we've always been troubled by that. In so much of medicine, we see the Hawthorne effect. If you look at the problem, then people all of a sudden feel symptomatically better. But whether they are having objective changes has always eluded us. So this is clearly an important thing.
Another point of this subjective issue is that people who doesn't see well in the periphery get very anxious. They are getting very anxiety. They're very fear about their not seeing, and not asking them whether they see it or not. By doing the objective test, help them better to do this test.
Tell us about some of your other work.
Since our care is to treat patients, so we have published last year a new treatment for retinitis pigmentosa patients. Those patients doesn't have any treatment actually, and they get more and more constricted visual field within the years, and eventually they get blind. We found with a special seaweed grown in the Red Sea. This seaweed has, if you grow it in particular climate, they have a lot of beta-carotene, but not like the carrot one. It is the 9-cis beta-carotene, which has a role in the visual pathway. The patients that we gave the 9-cis beta-carotene improved their vision, and at least one third of the patient did improve the vision in a crossover control mask study, and it was published in a very good journal.
Which compound do you think in this red seaweed is responsible for that improvement?
You're right. the seaweed contains a lot of compounds. We predict that the 9-cis beta carotene is the one who acts, but there are others, and may be helpful or maybe not. We'll probably eventually could tell, because besides that we're working on another project which is sponsored by Teva Pharma as a grant, and they are helping us to manufacture synthetically the 9-cis beta carotene, so we won't be needed to rely on climate to get the seaweed producing those good things.
Do you have a dose response curve for this seaweed product yet?
We used it in the same dosage as has been used for a decade, which found to be safe for other indications such as skin, psoriasis, and things like that. But since our budget is still limited, it's an agricultural product, we could not do still the dosage studies, but we surely would like to do that.
Is there any toxicity level?
The 9-cis beta carotene is less toxic than the vitamin A, because the vitamin A can do intracranial pressure. However, the beta carotene is a natural compound, and it actually does not do intracranial pressure.
Has anybody looked at beta carotene levels in the vitreous or in the anterior chamber?
No. It's the beta carotene does not add up even in the plasma, not add up in the blood. It accumulates in certain organs, for example, the liver, and probably the retinal pigment epithelium.
So have you done any studies to look at accumulation of beta carotene in the RP, obviously in subjects that have had this harvested for one or other reason?
Since our hospital is a large one, as I mentioned at the beginning, we have a large lipid center, and they are doing all the studies on the lipid mechanism and the beta carotene accumulation and absorption in mice and in human as well. Of course, as a blood test and things like that.
So ultimately, you see this particular preparation as being an important adjunct for prevention and retardation of AMD. Is that correct?
For AMD, we have not yet been done that. However, what we found in other retinal dystrophies or other retinal degeneration, that it did not just prevent it is improving the retinal function in patients with retinitis pigmentosa, at least for 1/3 of those patients that we started with.
And those are patients with what types of dystrophies?
Retinitis pigmentosa. We don't know the genetics. I'm not sure that the genetics is the only factor that is important for responsive patients. However, we will look at that as well in the future.
Is there anything else you'd like to add about any other projects you're working on?
I'm not sure we have the time.
Okay. Thank you very much, Professor. I appreciate it. And good luck, and we'll keep an eye on all the good stuff you're doing.
Thank you.
This is Dr. Dave Seftel reporting from Macular News, ARVO 2014.